Hello, hi, namaskara. My name is Kirtan and I am a second year medical student studying in a government medical college. Welcome to my channel Physiology at Once. Today I will be talking about the topic molecular basis of skeletal muscle contraction. So in this video I will be talking about the role of troponin, tropomyosin and calcium, the cross-bridge cycle, the role of ATP and cross-bridge cycling, and the types of contraction. So let's start this video. So before starting this video, if you haven't subscribed to my channel, please subscribe it because it's free and you can also support my mission of making physiology easy, simple and understandable. So let's start. So what happens in muscle contraction? There is sliding motion of the filaments. The filaments are the thick filament and thin filament, which I talked in the previous video. In the sliding motion of the filaments, it occurs due to the cyclic interaction between myosin cross bridges and the actin filaments in the presence of calcium and ATP. So calcium and ATP are required mainly for the muscle contraction process. So in the exam they may ask what is excitation-contraction coupling. So it is an important definition. So it is the phenomenon of pairing of electrical events, that is nothing but the conduction of action potentials through the T tubules, with the mechanical events. Mechanical events is nothing but the contraction process. It is known as the excitation-contraction coupling. So it is the pairing of these electrical and mechanical events. So what is the role of troponin, tropomyosin and calcium? So the troponin I and tropomyosin inhibits the myosin-actin interaction. I had talked about this in the previous video. So please check that video if you haven't watched it. I will provide the link of the video in the I button link. So the troponin T attaches the troponin molecules to the tropomyosin. So there are three troponins, troponin I, troponin C and troponin T. This is troponin I, this is troponin C and troponin T. This troponin T attaches it to the tropomyosin molecule. So this is a tropomyosin molecule. So this troponin I and tropomyosin inhibits the myosin-actin interaction. So it does not allow the myosin to attach to the actin. This troponin-tropomyosin complex acts as a relaxin protein that prevents the undesirable contraction. So for the contraction to occur, the Ca2 concentration increases in the cytoplasm. Then the calcium binds to the troponin C and induces a conformational change in the troponin molecule. So this results in the shifting of troponin I from the actin filament, and tropomyosin also moves away from the actin groove, resulting in the unmasking of the binding sites. So therefore, the myosin and actin are free to bind and form cross bridges. So therefore, it allows myosinates to interact with the actin molecules and engage in cross bridge cycling. So this cross bridge cycling is important in the muscle contraction process. So in the case of skeletal muscles, at a calcium concentration of 10 to the power minus 4 moles, there is full force development and the contractile system is fully saturated. So therefore, further increase in the concentration of calcium, there is very small increase in the force. So what does this mean? So the contractile system of the skeletal muscle is fully saturated at the calcium concentration of 10 to the power minus 4 moles. So when there is further increase in the concentration, so there will not much change in the force of the muscle. So whereas in the cardiac and smooth muscles, the amount of calcium regulates the degree of actin-myosin interaction. As in this, the contractile system is partially saturated. So when the concentration of calcium increases in the cardiac and smooth muscle, the force developed also increases. So let's talk about the cross-bridge cycle now. It is the sequence of events that occur during the interaction between myosin cross bridges and the actin molecules. So what are the events which takes place in the resting muscle? So in this phase, the ATP is attached to its specific binding site in the head of myosin, due to which the head is not allowed to attach to the actin. So as you know that myosin head contains sites for binding of ATP and actin. So when the ATP is binded to the head of the myosin, it is not free to be attached to the actin. So then the intrinsic ATPase activity of myosin head breaks the ATP to ADP and phosphate molecule. So after the breaking of the ATP to ADP and phosphate molecule, now the rotation of myosin head takes place. and the globular head is 90-degree angle in relation to the thick filament, pointing towards but not attached to the actin. So the head will be pointing towards the actin molecule but not attached to the molecule. This conformation is known as the energized state. So when most of the myosin molecules are in the energized state, it is the relaxation phase. So now let's talk about what are the events which takes place in the stimulated muscle. So when the action potential arrives, the cytoplasmic calcium increases and the myosin binding sites on the actin are exposed. So as you know, when the calcium increases in the cytoplasm, the troponin C binds with the calcium and it makes the tropomyosin and troponin complex to detach from the actin, and therefore the myosin binding sites on the actin are exposed. So after the exposure of this myosin binding sites on the actin, the myosin ADP phosphate complex has high affinity for actin, and therefore the energized myosin head binds to the actin, forming the actin myosin complex or cross bridge formation. So this is the cross bridge formation process. So what happens in the cross bridge? It has two effects. First, the phosphate molecule is released from the myosin head, producing a power stroke. So power stroke is a conformational change in which myosin head bends at an angle of 45 degrees, pulling the actin filament towards the center. So here it bends at 90-degree angle in the resting phase of the muscle. So but in the stimulated muscle, the myosin head bends 45 degrees and attaches to the actin filament and pulling it towards the center. Secondly, the ADP dissociates from the myosin head, leaving the actomyosin complex in a rigid state. So what happens after the ATP dissociates? The myosin head becomes rigid and stays in the attached position to the actin. So when the ADP-free actomyosin complex attaches to the ATP, resulting in the detachment of cross bridges from thin filaments called at discharge state. So when this ADP-free actin-myosin complex attaches to ATP, there is detachment of the cross bridges from the actin filaments. So this is called as the detached state. Soon the ATP hydrolysis occurs and the process again continues. The myosin head becomes energized and the cycle continues again. So what is the role of ATP in the cross bridge cycling? It is mainly required for the detachment of the myosin head from the thin filaments, as you saw now. So what is the clinical significance? When cellular ATP stores are depleted, it actually happens after death. Cross bridge cycling cannot detach, and this causes the thin filaments to remain in attached position, and hence the stiffening of the muscle. So when the thin filaments remain attached to the myosin molecule, there will be rigidity of the muscle and cause stiffening. This phenomenon is known as the rigor mortis. So it is a main sign of death. It actually occurs between 4 to 12 hours of death. So let's revise this cross bridge cycling with a small diagram. So this is the energized state. The myosin head contains ATP and phosphate molecules and is directed toward the actin filament, but it is not attached to it. So afterwards there is cross bridge formation due to the increase of calcium concentration in the cytoplasm. So the myosin head attaches now to the actin filament with the ADP and phosphate molecule attached to the myosin head. So afterwards there is detachment of the phosphate molecule and slowly the ADP molecule also, which is called as power stroke. So due to this power stroke, the actin molecules are pulled towards the center and there is the shortening of the sarcomere length. The A band remains unchanged, the H zone and I band are shortened. So after the power stroke, there is attachment of ATP to the free myosin head, due to which detachment occurs and it leaves the actin filament. So then the cycle continues again. So this is the flow chart to remember simply. So there are 4 main steps: ATP hydrolysis in myosin head, which causes energized state; cross bridge formation in the presence of increased calcium; release of ADP and phosphate molecule from the myosin head, which is nothing but the power stroke, then the binding of ATP to myosin head, ultimately causing the detachment, and this process takes place again. So now let's talk about the types of contraction which takes place in the muscle. So there are mainly two types of contraction, isometric contraction and isotonic contraction. This also may be asked in the board examination for 3 marks. So in the isometric contraction, there is no apparent change in the muscle length. So therefore it's isometric. Metric means length. So what is the example? When you try to lift a heavy object or pushing a wall. So when you are trying to lift a heavy object, there will be no muscle length change, but you will still be exerting the tension. The cross bridges bind with the actin but unable to drag them because the load is greater than the tension. The load or a heavy object is greater than the tension applied by the muscle. So therefore you cannot do the external work in this case. So since no displacement occurs, there is no external work. So in the case of isotonic contraction, there is no apparent change in the muscle tone. The example is lifting an object, walking, running, etc. So while you lift the object, there will be change in the length of the muscle, but the muscle tone will be same. In this, the cross bridges drag the actin filaments because the tension exerted is greater than the load. So you can easily carry the load or pick it up. So since there is displacement in this case, the external work is done. So that's it for today guys. If you found this video useful and informative, please like, share and comment, and also don't forget to subscribe. Thank you.